EP 120, Chapter 9, Topics, Types of Neurons and at the Synapse. All right, we can distinguish neurons by their function as well as by their structure. So when we talk about neurons by function, the first kind are the sensory neurons. Sensory neurons detect changes in the environment via their dendrites, or there's a separate receptor cell that is detecting sensation and sending the information to the sensory neuron. But the key thing is a sensory neuron is sending information to the central nervous system. So from the body to the central nervous system, spinal cord or brain. That's what sensory neurons do. And all sensory neurons are unipolar and house their cell bodies in the dorsal root ganglion. Then, within the central nervous system are the interneurons. Interneurons are only found in the central nervous system, and they receive information, process the information, and then send out impulses, send out commands. So interneurons are what make up the brain and spinal cord and receive, process, and respond to information. And they are always multipolar. And finally, we have the motor neurons. Motor neurons control the effectors, so they control the muscles and muscle tissues and also glands. And the motor neurons, their key thing is they carry information away from the central nervous system and to the effector. So again, motor neurons carry commands, impulses from the central nervous system to the effectors in the body. And motor neurons are multipolar. So this division of the nervous system by the function of neurons is also how we can divide it by the function of the nervous system as a whole. So peripheral nervous system, sensory receptors, sending information to the sensory neuron, basically the receptors detecting changes in the environment. Sometimes the sensory neuron is the sensory receptor. And then they send information to the central nervous system, where interneurons will process and integrate that information and then send out commands via the motor neurons to the effectors. Nuclei. Here's another vocabulary term. Nuclei are specialized gray masses found in the central nervous system. Since they're gray, that means there are lots of neural cell bodies in these locations. So nucleus is a gray mass within the white matter of the central nervous system, composed of neural cell bodies. All right, so hopefully you remember what these three different kinds of structures are called. Boom, I hope you got it right. All right, where are ganglions found? Where are nuclei found? Boom, I hope you remembered. Ganglions are in the peripheral nervous system, nuclei in the central nervous system. And what are they made of? What composes these structures? It is neural cell bodies. So ganglions in the peripheral nervous system, high concentration of neural cell bodies. Nuclei or nucleus in the central nervous system, high concentration of neural cell bodies. All right, returning to the idea of the synapse. So again, the synapse is a functional connection between two neurons, or a neuron and an effector. There's a space between the two cells called the synaptic cleft, and the goal is to send information from the presynaptic neuron across the synaptic cleft to the postsynaptic neuron. So how does the message cross the synaptic cleft? How does it get past that thin little space? Here's the options. Hope you got diffusion of neurotransmitters. So the synaptic knob releases neurotransmitters that diffuse across the synaptic cleft to the next cell. So here we have the very end of an axon for a presynaptic neuron. The very end of the axon is the synaptic knob. Synaptic knob has many vesicles, many synaptic vesicles. And within these synaptic vesicles are neurotransmitters. The synaptic knobs at the end of synapses, at the end of axons, possess the synaptic vesicles filled with neurotransmitters. So a signal, a nerve impulse arrives. It triggers the synaptic vesicles to undergo exocytosis, to migrate to the surface of the synaptic knob, fuse with the membrane, and release the neurotransmitters into the synaptic cleft. Neurotransmitters then diffuse across the synaptic cleft and bind to receptors on the postsynaptic neuron. And these receptors are part of ligand-gated ion channels, so that when the neurotransmitter binds to the receptor, an ion channel will open, letting ions flood into the postsynaptic neuron. So that's the basic story of a synapse. And it's worth noting that the neurotransmitters can be excitatory, can lead to another nerve impulse being generated in the postsynaptic neuron, or it can be inhibitory and prevent the formation of another nerve impulse in the postsynaptic neuron. Here's just a electron microscope view that's been colorized by a computer. Here is the synaptic knob with all those synaptic vesicles. Here is the postsynaptic membrane. You can still see the synaptic cleft. Pretty cool. All right, neurons. Turns out that the cell membrane on neurons are polarized, which means that it has a charge. Specifically, there is a slight negative charge on the inside of the neuron and a slight positive charge on the outside of the neuron. And this charge, this polarization, is necessary so that neurons can generate nerve impulses and generate what are known as action potentials. And the charge in a resting neuron is negative 70 millivolts. So the resting membrane potential is negative 70 millivolts. Remember that. So it turns out that it is the distribution of sodium ions and potassium ions that help maintain this polarization. So you have a high concentration of sodium ions on the outside of the neuron and a low on the inside. You have a high concentration of potassium ions on the inside of the neuron and low on the outside. And this gives us an overall positive charge outside of the neuron and a slight negative charge inside, partly because of other things inside the neuron that are negatively charged. Now, it turns out that the neuron is constantly leaking sodium and potassium. So it constantly has sodium entering the neuron and potassium leaving the neuron. And this, if left unchecked, will disrupt that balance, will disrupt the polarization. So there is a specialized protein that will help pump sodium ions out of the cell and pump potassium ions back into the cell, called the sodium potassium ion pump. It is a transmembrane protein that is engaging in active transport, so it needs energy. It needs ATP to move these ions. And to be honest, the reason why our nervous tissue is so high demand for energy, why it needs so much energy to function, is to maintain this distribution of ions through the function of the sodium potassium ion pump. Now, the sodium potassium pumps will move 3 sodium ions out of the neuron and 2 potassium ions into the neuron. So each single ATP that is expended causes 3 sodium ions to leave the neuron and 2 potassium ions to enter. And that is where we'll stop with this part of the lecture.